I've just picked a topic today a little bit different to what most people might talk to you about when it comes to animal welfare, but quite often we measure hormones when we're trying to assess welfare, and we often try and associate stress with that. So I'm just to talk a little bit about that, but before I do, I just want to make an acknowledgement to the fact that the research, the body of research that I'm going to refer to has been long in the making, decades, and many people have played various roles towards the research area. One of my hallmarks is collaboration. I believe strongly in it. I've got quite a lot of collaborative links nationally and internationally that I think are very important in terms of taking research forward, and likewise have a fairly broad funding base that goes behind a lot of the work that I'll talk to you about. And I think it's important to be able to tap into the different funding sources depending on what's required for the time. So quite a lot of people have played various roles in the general area that I'm going to talk to you about. When we think about hormone stress and animal welfare, we're actually trying to put together some very complex and challenging areas that are difficult for a range of reasons, and one of them is that it's difficult to define animal welfare. It can be a subjective term, we've all got different views. I'll talk to you a little bit about that. Likewise, defining stress is also a very difficult thing to do, and again, we tend to have our own views on what stress is. Another challenge is that the endocrine system, the system that produces the hormones, is really complex, and I've been working on it for a long time and still don't understand it. And then trying to associate stress and welfare becomes a huge challenge because, it's, as you'll see in a moment, they're not necessarily clearly linked. What's really important, the important message to take away, even if this whole streaming thing was to fall down now, the lights were to go off, and we all had to run out because there's a fire alarm, is that what we need to know are the consequences of the actions of hormones. It's the actions of hormones is where a lot of the research has stopped. It's actually what happens as a result of those actions that become important in terms of trying to assess the welfare of animals. We'll come back to that. Animal welfare is pretty difficult to define. In terms of a research pursuit, it's quite a young area. It probably kicked off properly in the mid-1960s with the Bramble Committee that was commissioned by the UK government to look into the way animals are handled and the way we should handle animals, and out of that was spawned the 5 freedoms, which you might have heard about, and there was a whole bunch of other different ways to define animal welfare have come from that, such as the 5 domains, animal and life's worth living, the Welfare Quality Assurance Project in Europe, and so on. But of all of these, there are 3 concepts that are probably becoming the most accepted in terms of trying to define animal welfare. One of these is called the naturalness concept, which is the animal being able to exhibit natural behaviour. From a research perspective, we find that fairly unacceptable because it's so subjective and so difficult to define. But another concept that is important is the biological functioning concept. This is where stress does become important, because what this is about is whether or not an animal's body is able to adapt and be able to respond to challenges in a positive way to allow it to function normally. And a more recent, and perhaps up until very recently controversial concept has been the affective or emotional states framework of animal welfare, and it is now generally well accepted that animals, our livestock, are sentient organisms. They do have emotions, and in addition to being able to see how they function from a physiological perspective, we also need to consider how they function from an emotive or affective states perspective as well, and that's difficult. That's a really difficult area and challenging area to actually do research on. Stress, a really difficult area to define. It's a much older area of pursuit than is welfare. Probably where stress really started to get its grounding in terms of having some concrete definitions around it was the early work of Bernard, who was really instrumental in showing us that cells needed to have an internal stable environment in order to operate normally. And when that was upset, then the cell function was upset and it needed to be re-established in order to get back to normal. And that's where Cannon, the US physiologist, was very important. He's the one who actually developed the homeostasis philosophy that's very important to physiologists around the world. And in particular, his work put the sympathoadrenal system as a frontline stress responsive area on the map. And of course, he's the guy who coined the fight or flight syndrome. Now, the sympathoadrenal system is really important in dealing with acute stress responses, so ones that don't last long. Its components include the brain, with direct innervation from the spinal cord of various tissues, also direct innervation of the adrenal glands, where you get the release of catecholamines. So you've got the epinephrine and norepinephrine there, which is adrenaline and noradrenaline, released into the blood and travelling around the body, having effects, all sorts of different effects on the body. And a hallmark of this system is that it responds quickly, its actions are fast, and the actions dissipate quickly. And this is just some data from sheep that were stressed here, and you can see increase in adrenaline responses, very, very rapid increase, very, very rapid dissipation. And that's standard sort of profile of catecholamines when an animal's stressed. And this is where we can start to think about where the challenges come in trying to understand hormones and stress and welfare. If we have a look at this photograph here, both the guys and the bulls have got similar activations in terms of their sympathoadrenal system. There's no question that these guys are definitely in a flight mode, but what about the bulls? Are the bulls in a fight mode or are they in a flight mode? Either way, they've got lots of adrenaline floating around their blood. Then where the progress really came was from Hans Selye, who did his seminal work in Montreal in Canada. And it was Hans Selye who developed the general adaptation syndrome, and in particular highlighted the importance of the hypothalamo-pituitary adrenal axis in dealing with stress. A really, really important physiological system when dealing with stress, and of course when taking that on further to understand welfare. Basically, the higher neural centres detect challenges, which we tend to call stressors for easiness, and then it stimulates the release of corticotrophin-releasing hormone and arginine vasopressin into hypophyseal portal blood systems. That drives the production of adrenocorticotrophic hormone, which drives the action of the adrenal glands, and in particular to produce glucocorticoids. And the one that's the main glucocorticoid in most mammalian species is cortisol. It's corticosterone in a bunch of rodent species and in avian species, but cortisol tends to be the hormone that most of us measure when we're looking at stress and indeed welfare. And cortisol has very large number of actions throughout the body, and one of the challenging aspects of cortisol is it's not just a stress hormone, but it does lots of other things as well, and I'll come back to that point. Unlike the sympathoadrenal system, here we've got cortisol concentrations in the same sheep stressed, and you can see the concentrations go up just like adrenaline did, but here the cortisol takes much longer to dissipate. So cortisol is a steroid hormone. It's not stored, it's synthesized when it's required, and it takes longer to act, and that becomes important when trying to interpret the results. We've tried hard to develop a definition of stress that stood the test of time. This is what we came up with in the mid-2000s. It's a complicated physiological mechanism that embodies a range of integrative physiological and behavioural processes that occur when there's a real or perceived threat to homeostasis. To be honest with you, in the 11 years since we coined that, this hasn't helped us much either. Still a lot of words, and it's not that simple, even though there's a lot of words there. But when we start to think about what actually happens when an animal is stressed, and then try and relate that to welfare, let's just start from the beginning and think what happens in terms of the changes in hormone synthesis and secretion. And I've put the catecholamine, so the sympathoadrenal system and the HPA axis up there. I've just popped the reproductive axis in, you'll see why in a little while. And what happens is that all of the things that are released from the brain happen quickly in the HPA axis. Catecholamines are released instantaneously. Catecholamines are synthesised and stored, so they're sitting there in their cells ready to go. But the glucocorticoids like cortisol, they take some time before their concentrations go up because they have to be synthesised. They're not stored in the first place. All these things matter when it comes to interpreting what hormones are doing and how it might impact the body of the animal. So if we then go to the target cells where these hormones act on, we'll find that there are that things happen in different time frames as well. And these time frames are important, again, when interpreting what happens. So the glucocorticoids, for example, take a while to be made before they go up, and then they take a bit longer to actually have their actions as well, and that's because they have to interact directly with DNA and affect transcription, whereas the catecholamines will have a very fast effect because they're going to act on second messenger systems in cells and release things immediately. We'll come back to that. But this is where it gets really important. Remember, the take-home word is consequences. It's all very well being able to measure hormones and say this is what happens in target tissues, but what are the consequences from a physiological and a behavioural perspective on the animal? And here's an example of some of the things that happen over time when a stress is imposed. And these are just a handful of things that have happened. You get changes in cardiovascular tone and blood flow and appetite and all sorts of things. The key question though is, when is the animal's welfare impacted? When do any of these things here matter in terms of how we might assess the welfare status of the animal? And here I want you to think not just in terms of welfare being a negative thing, which is what most people do. Well, think, well, the welfare of an animal is bad, where there's actually now a push for us to appreciate that to not only move animals from a negative state of welfare, not to one that's acceptable, but indeed to one that's positive. So when do any of these things that are happening here actually mean that the welfare is impacted? And it's complicated. For example, have a look at the immune function here. If we have a look at the effect of stress on the immune function, initially there's an activation of the immune function. That's a good thing. And then there's a suppression of the immune function. So initially when we have the onset of stress, we get this increase in immune function. That's to help you fight the challenge that's come your way. But if it keeps going, if there's a constant increase in cortisol concentrations all of the time, then what happens, of course, is the immune function starts to decrease and can be completely and absolutely inhibited. And indeed, we use that in a medical sense to our advantage at times, to decrease inflammation, for example. But if an animal's in this chronic state of immune suppression for a long, long time, its welfare might be affected. And one of the studies that stood the test of time in pigs is this work that was done by John Barnett 25 years or so ago. And this shows the concentrations of cortisol on the left-hand panel and immunological responsiveness on the right. And what happened was he had pigs and he just they had plenty of space, and then he cut their space down. So he confined them, in other words. It put cortisol up. Now this is over a very long time. This is over many, many, many weeks. So here we're talking about a chronic treatment. Over many, many weeks, cortisol goes up, immunological responsiveness goes down. Now there's no cause and effect described in this study, but it seemed to be that most people have accepted over the last 25 years or so that that's pretty reasonable evidence that when you're stressing an animal for a long time, which you can measure through cortisol concentrations, then it'll suppress its immune system, and its welfare is probably going to be, in this case, negatively affected. But it's never quite so clear. Have a look at this study we did, admittedly over a much shorter time. This shows concentrations of cortisol, and this is in sheep. And the black bar here represents when the sheep were stressed. Now at point 0, the sheep were fed. So came in the morning, fed the sheep, and then took blood samples every 10 minutes for all of that time there. And you can see that in response to feeding, the cortisol concentrations have gone up, and indeed they've gone up at least as much as those concentrations during stress, and they probably stayed up a bit longer as well. So you've got as big an effect on cortisol concentrations through feeding as you have through imposing a stress. And so if you were just to measure these concentrations and try and interpret from that, you could be in a little bit of strife. Because here, as an example, we measured luteinizing hormone in these animals, And down the bottom we've got the pattern of secretion of the luteinizing hormone, and it typically has a pulsatile pattern of secretion. And this is what happened. When the concentrations of cortisol were high due to feeding, LH was fine, no problem at all. But when it was high due to stress, it was suppressed. So cortisol did different things under those two different circumstances. Now, often we use this LH as a barometer of physiological function. I'm not saying that the welfare of the animal's been impacted here. That's not the point. The point is that the high concentrations of cortisol are doing different things in different settings. And here, cortisol is probably not acting as a stress hormone, but as a counter-regulatory hormone to counter the actions of insulin from the feeding. Whereas here, it's acting as a stress hormone. So again, it throws open just how complex the whole system is that we're dealing with. It's not just a question of being unable to have straight relationships between stress and welfare, but the hormones that you use to measure that or to assess that are really a complex system. And the reasons are, there's lots of systems. There's not just the HPA axis and the sympathoadrenal system. There's lots and lots of other hormones as well. Some of the hormones are synthesised when they're required and stored, and some are synthesised and not stored, so it can take longer. Some hormones are not secreted, actively secreted. Glucocorticoids are an example of that. They leave the cell passively. It takes them longer to get out there and do things. Catecholamines leave quickly. Some hormones like the cortisol needs to be transported in the blood. That takes longer. It's got to associate with a protein, be carried around in the blood, dissociate, get in the cell, whereas other ones like the catecholamines can flick around quickly. They vary in their actions. I want to make a point on that in just a moment. And it's the consequences of these actions that become very, very important when it's coming to assessing welfare. And of course the hormones are all metabolised differently at different rates, and they'll sit in tissues to different amounts of times as well. When it comes to action, we've got two types of actions that hormones that can have. Some will bind to receptors and stimulate second messenger systems to influence the function of proteins. That's rapid. That happens very, very quickly. Others need to actually interact with DNA to influence transcription. And if we're going to actually understand the consequences of the actions, we have to climb inside the target cells and understand what's happening in there. And some of our research has shown that it's actually not often enough to just measure the hormones in the blood or any other external medium for that matter. You actually need to know what's happening in the target cell before you can get a handle on the consequences of the actions, which may or may not influence the welfare of the animal. And now, just to add to the complexity, all of these hormonal responses vary. There's rhythms in their action. The type of stressor and the sex of the animal can have an effect, and the physiological state of the animal can all influence responses to stress, which can then influence the impact on welfare. When it's well known, absolutely well known, that there's diurnal variation of stress responses. This is sort of cortisol concentrations. They all vary. They vary in different amounts in different species, but they vary. And depending where you are in the cycle, where you're taking your measurements, where you're imposing your treatments, where you're transporting your animals, where you're feeding your animals, can have an effect. The other areas, the other thing, add the complexity, the type of stressor and the sex of the animal. For example, if we consider one type of stress, which sheep, a very good stressor in sheep, isolation and restraint, you take the sheep from its friends and you put it in another room and you restrain it. They don't like that, and they have a stress response. And this is an example of cortisol concentrations in males and females that have undergone this isolation and restraint stress. And the cortisol concentrations in the females are typically higher than those in the males. Now, it's hard to interpret that. The females have had a greater stress response in this setting. Does that mean that they're worse off or better off? Are they better able to cope with this challenge, or are they more stressed and so therefore they're going to be worse off? Now, another type of stress, a predator stress. You bring in the dog whose name is Molly, and she barks at the sheep, and they don't like that. And here you see this is where the dog barked. Concentrations go up of cortisol, much higher in females, and that's a very repeatable response. So just like in isolation and restraint stress, the females and the males will have a different response. But if you turn to, say, a metabolic stress and give a shot of insulin, the cortisol concentrations go up higher in the males. Is that a stress response or a counter-regulatory response? Either way, it's a sex difference, and it needs to be considered when we're looking at what's going on. The amount of visceral fat on our animals, ruminants included, will influence their stress responsiveness. For example, here we have stress imposed here, cortisol again, much higher in animals that have got very high levels of visceral fat through dietary feeding. We put these animals on planes of nutrition to get particular levels of fat in there, and a much higher concentration in obesity. So the physiological state becomes important. Another physiological state that's important is lactation. For example, if we have a look at the top panel, non-lactating animals, the bottom panel's lactating animals, this is where the stress was imposed. The round circles is the day where the stress was imposed. Normal cortisol response in your non-lactating animals. Lactating animals with their lambs present, no responses at all. So the lactation has wiped out the ability of the HP axis to respond. Or has it? Have a look here. This is a study that we just published recently. On the left-hand panel, that we've got lactating ewes with their lambs present, and this is the isolation and restraint stress, which I showed you before, no responses effectively. But what happens when we introduce barking dogs? Suddenly these lactating ewes that didn't have a stress response to isolation and restraint had these stress responses. They needed to have stress responses in order to protect their lambs. So the setting's different. Are these animals worse off or better off? Certainly the lambs are better off because the ewes become aggressive. So again, highlighting how complex it is to interpret endocrine responses in terms of physiological welfare. So one of the things that we've recently proposed is to learn from our studies in reproduction and impose those learnings on welfare. We're suggesting a case study where we apply the approach that we've done in reproduction on animal welfare. Let me explain it to you. Over a period of about 12 years or so, we undertook a major program which was looking at insights into how stress affects reproduction in females and are glucocorticoids involved. In other words, we know stress suppresses reproduction. How much of that's due to cortisol? Once we know that, then we can go into a particular setting. We know that when we measure cortisol in this setting, we know what it does. We know that it's bad or it's good or it doesn't matter. We want to be able to achieve the same thing with the hormones in welfare. These studies were done on sheep where we looked at stress responses in sheep, and we had a focus on the hypothalamo-pituitary gonadal axis, which releases gonadotrophin releasing hormone, drives the anterior pituitary to produce the gonadotrophins, which are important for gonadal function, gametes and hormones, and these hormones have important effects. So we undertook a series of studies that will allow us to dissect this axis at different spots and focused on the production of luteinizing hormone and on sexual behaviour. So if we just go to this slide, which has got all the endocrine actions that we know about in female livestock, all these hormones are doing these things. In addition to that, there's a period where the sexual receptivity will take place, the estrus period. There's different sorts of sexual behaviours, quite complex, attractivity, proceptivity, receptivity that happen in there to allow the animals to get pregnant or not. And we know, and everybody knows from many, many years of research, that stress inhibits all these things. Okay? Stress inhibits all of these things in there. But what the question here is to what extent is cortisol responsible for these effects of stress? You know, try and translate this to welfare. This is reproduction, but if we're not at this stage yet, we haven't started, but if we were at the position we are here and we've done the research, we would know what impacts cortisol will have on these particular physiological and behavioural functions that influence welfare. But here the focus was reproduction. These effects of stress, there's lots of them. They all have inhibitory effects on reproduction, but the focus here is going to be the psychosocial stressors. And the approach that we used was a bit similar to the isolation and restraint stress I showed you before, except we added a couple of extra ones, blindfolding the sheep and bringing in a barking dog, and we called it the layered stress paradigm. And over a series of many, many experiments, over the better part of a decade, we were able to demonstrate that if you put in the layered stress paradigm, you decrease the synthesis and secretion of gonadotrophin-releasing hormone from the brain. That's shown by these much smaller pulses of GnRH here. That you decrease, you suppress LH secretion from the anterior pituitary. And here's just proving that cortisol goes up. But the question is, to what extent is cortisol responsible for these effects of the stress on there and on there? And cortisol, if cortisol is having an effect here, it could be through suppression of GnRH. If it's having an effect here, it could be by having direct actions on the anterior pituitary or in the brain or both. And over a series of crucial experiments where we interrogated the actions of stress and hormones throughout this axis, we were able to show that yes, when we impose this particular stressor, cortisol has direct actions at the pituitary gland to suppress the output of luteinizing hormone, and we've gone on to show what the cellular mechanisms behind that are. But no, the effects in the brain of the stress are not due to cortisol, they're due to something else. And we've been doing some work on what that something else might be. That's not the point for today. The point for the day is that measuring the cortisol on its own without this sort of knowledge is of no use to us in terms of taking it to the point where we want to know what the consequences of the hormone's actions are. We now know that the consequences of the hormone's actions are to have an effect at the anterior pituitary, but not at the brain. And what we want to show, we want to take this same approach into the sphere of welfare so that we can understand where our hormones have actions or not that are important for welfare. Same thing with behaviour. There's 3 types of critical sexual behaviours. You don't need to worry about what they are for today. The point is they're all suppressed by stress. The top one's not due to cortisol, the bottom one's not, but the middle one's not, but the bottom one is. So in other words, same again, cortisol, we can put the animals in. We know in the setting which setting cortisol is going to actually be responsible for inhibiting sexual behaviour. We want to get to the same point with welfare. So the point is, how useful are measuring hormones to understand animal welfare? How useful is it? It's done all the time. It's done mostly because people like to link stress with welfare. But it's not that easy. The endocrine system's complex, the responses are complex, the consequences of the responses are complex. Just to recap and finalise off, stressors, of which there are many and varied, will have an influence on the synthesis and secretion of lots of hormones, not just the 2 standard frontline stress systems that we know about, but lots of hormones. These hormones have actions, they vary heaps depending on the nature of what hormone they are. And you'll have the same hormones having different actions at different parts of the body at the same time. It's the consequences, the physiological and behavioural consequences that we need to understand before we can go forward and use those measurements in assessment of animal welfare. And we have reached a point now where the livestock industries in this country, particularly the red meat industries and the pork industry, have determined that we must continuously improve animal welfare, or we are looking at market failure. And the only way that we can continuously improve animal welfare is to have a very robust, repeatable, straightforward means of assessing the welfare of animals. Thanks.